Okay, so we were discussing science and technology in the current affairs series. Today, we also continue with that. So we have some more topics. So today to be discussed. So there were many fusion reactors which were in news. Even in 2024, we had many in news, especially in UK. Okay, so we had it in Korea, we had it in France, right? So many other areas. So fusion reactors, high temperature reactors, okay, all these reactors were in news. Apart from that, we also had some news about nuclear plants in India, okay, fission reactors. So apart from that, we have hemoglobin bodies, Casgevy, and CART cell therapy and funga. So these four issues we will discuss at large, and we will see to it. So in that, the first we go with fusion reactions. Okay. So what is fusion reaction? Why we are much into fusion reaction? So what is the purpose behind it?
The fusion, the amount of energy released is comparatively higher than fission. So we are much obsessed with fusion.
Okay.
So security purpose also, like it means like it is more secured, and like it cannot like if the certain conditions are not fulfilled, it will not work. So this
Okay.
is the one of the main reason that we are
Okay.
looking for the conditions.
Fine. Then, so it gives more energy. Fine. Okay. So what difficulty we have in fusion reaction?
So to achieve that particular temperature, to maintain that temperature, to make it self-sustainable is not an easy task. Means like right now what's happening, that like the amount of energy that we are giving is more compared to the energy that we are receiving. So it's not sustainable, feasible. So
okay. So as of now, the technology is that, see, generally to happen a fusion, we need to have a fission first. And with that energy, we try to have the fusion, and that will be sustainable where we try to get that particular thing. So getting criticality in fusion reaction is difficult. So that is a problem what we have. Okay. And this particular reactors we call the fusion reactors, which we have in the earlier days. So we all started with the International Thermonuclear Reactor or Thermonuclear Energy Reactor, ITER. So that is a first largest step what we have been with international level, and later there were many tokamak which came in. Okay, so we'll see one by one in that. Okay, so why you call it as tokamak? Any idea? Okay, so it is Japanese or Chinese word. Okay, fine. Then any other idea? So it's a Chinese or Japanese term as you mentioned. Okay. Then so actually, tokamak is not a Chinese or Japanese term. It is actually a Russian term. Okay, so remember that. That's the reason why I asked why we call it as tokamak, because you will fall for it as you know Korean or Chinese or certain things. So we call it as tokamak because of the Russian acronym, okay, toroidal chamber with magnetic coils. So that we call it as tokamak, okay, Russian term. Fine. Anyway, so what happens here? So generally, this particular the Japanese tokamak, which was in news, so they became the world's biggest experimental nuclear fission reactor. So which could generate large scale energy at and the SA stands for super advanced, right? So the main purpose of this is to replicate that particular nuclear fusion ignition. That is the net surplus energy. We try to have it, and that's the main criteria what we want to have, and that is where the idea come into this uh, uh, existence. Okay, so that is where we have this particular aspect. Right. So the net energy gain what we have in this, so that has to self-sustain. So that's the second part what we have, which becomes even more important. So what is fusion? So fusion is two small or light atomic nuclei joining or fusing into a single heavy atom or nuclei. Whereas fission, so it is you divide a particular heavy atom into two. So that we call this fission. So fusion, fission. So this particular fusion reaction needs energy to start up with. So that is a greater challenge what we have. Right. So we try to see this to come up. So Japan. So try to remember that. And similarly, we had the earlier one, which was in France, that we called as ITER. Yeah. So whether India had tested nuclear fusion? Whether India has tested nuclear fusion? Thermonuclear device. So we have done. Pokhran 2 is that, right? So Pokhran 2 is thermonuclear device. So which is nothing but a fusion reaction test. Anyway, so check up with nuclear fusion. So why nuclear fusion is difficult and why it is important, and what are the various other important features about attaining criticality in a nuclear fusion reactor. So that is important. And apart from that, related to these nuclear reactors, so in India also there are many nuclear reactors which were in news. So especially the fast breeder reactors. So what are fast breeder reactors? What are fast breeder reactors?
The fast breeder reactors, like the amount of fuel that it consumes, it it produce also like more fuel for the further uses, like fissile and like fertile. So basically, it converts a fertile fuel to a fissile.
Okay,
So the
so
amount
it is
of
a
fuel,
breeder reactor. Breeder reactor means it generates its own fuel, right? So
yes,
do we have a fast breeder reactor?
so like recently I saw a news where like we have achieved something in fast breeder. We are working in some, like actually I'm not remember the place, but
Anybody else? FBR.
Yes, so we do have.
Okay, what is the name of it?
Like it's called prototype fast reactor, right? PFPR.
Or fast breeder test reactor. Okay. So where it is? Kalpakam, it is called as Bhavani, right? So Bhavani installation has that particular separate. Bhavani is a atomic power station which is there in Kalpakam itself, which is having that prototype FBR. Okay. So recently it got the criticality level, right? Fine. So that is important. Apart from that, advanced heavy reactors. So heavy water reactors. What are advanced heavy water reactors? What is the difference between these two? FBR and advanced heavy reactor. Advanced heavy reactors are those which can use thorium. So thorium partially, a mixed carbide fuel will be there. So thorium will be mixed into that particular reactor. So from thorium, the disintegration can be partially started because of the mixed carbide fuel. So from that, the output will be there. That output, plutonium, can be used as an input to FBR. And similarly, uranium-233 can also be there. That can also be utilized. Okay, that is the capability of advanced nuclear reactor. For that only, Kalpaka mini reactor or Kamini, which operates in uranium-233, will be there. So another important factor in FBR, that is fast breeder reactor, is that apart from other reactors, fast breeder reactors lack what? What you actually lack in fast breeder reactors? In other reactors, you find it, whereas in fast breeder reactor, you don't find it. What you won't find?
Moderator.
Moderator, right? Good. So moderator will not be there. Why? Why you don't have moderator in fast breeder reactors? Because it is a fast breeder reactor. So you need to have fast neutrons. The 3 neutrons will be there and you will not moderate the particular 3 neutrons to 1. You will not absorb it. Why? Because the fast breeder reactor has to have the 3 neutrons which are fastest hitting that particular core. So it has to work. So then only the fast breeder reactor can involve in the chain reaction. So otherwise it won't be a fast breeder reactor. Whereas in other reactions, we don't need that particular fast breeder. It's not a breeder reactor. So whether it is a pressurized heavy water reactor or advanced reactors, we have moderators. Clear? Okay. So certain areas which we have, say Kakrapar, the single largest unit, what you have. Okay. So remember, Kakrapar is a single reactor, largest reactor, we have it there. Largest nuclear plant, you have it in Kudankulam. So already we have about to be constructed and it is going to be expanded to 8. So almost the larger reactor you have it in, the plant you have it in Kudankulam, whereas single plant which is largest, it is going to be in Kakrapar. So these are certain areas where you had it in news, okay, with related to nuclear power. So remember that. Fine. So then we move on to the hemoglobin body, the Casgevy, as well as CART. So all these things are related to each other. Okay. So what is hemoglobin?
So the red blood cells present in blood or hemoglobin.
Okay.
Pigment in the blood.
Pigment in the blood, we call it as hemoglobin. Fine. Okay. So what is the purpose of hemoglobin?
They carry oxygen.
Oxygen carrying.
Okay, they carry oxygen. Okay, fine. Why they carry oxygen?
Because it needs to be transported to all the organs.
How it transports? That is, how it can carry oxygen. When other elements couldn't, why it could carry oxygen?
Sir, maybe it can form bonds with the oxygen.
Okay. So which can enable it to form that particular bond?
So iron.
Iron, right? So one of the major feature why hemoglobin is associated with that oxygen is that it is iron. Okay, so, fine. When you lose that particular iron, so what will happen to hemoglobin? When iron is not present in hemoglobin,
It cannot absorb and transport oxygen.
so it cannot absorb or transport oxygen. And that particular area, we have the problem. So this hemoglobin, which doesn't have that particular capability of transportation because of oxygen, deficiency of iron, that leads to many issues. So one such problem is that what you can see, anemic condition. So you call it as anemic conditions where you don't have iron. So rich iron rich hemoglobin will not be there. So naturally there will be a problem. Okay. So hemoglobin is an important component which results in many or impacts many functions, not only in the metabolism or the very existence of the organism, but also its role played in disease control or in disease is very significant. And that is the reason why we always have that particular habit of checking the blood samples. So what type of issues we have it in the blood and so on. So in this background, usually, so what is the source of hemoglobin? Generally, hemoglobin means immediately what you will see. What is the source? From where hemoglobin generates? From where blood generates in human body?
Sir, red
Joy.
blood cells.
Bone marrow.
Bone marrow. Okay, so bone marrow. Bone marrow is a general known source of hemoglobin, right? So bone marrow is a place where hemoglobin is generated. Okay. So now scientists have reported that hemoglobin has another source. Okay. So another source, what they have seen is chondrocytes. Okay. So chondrocytes. So they also have what do you call the ability to carry that particular oxygen or oxygen reservoir, which is also one of the area where hemoglobin is present in general. Clear? So that is one of the significant discovery what we had made. So remember that hemoglobin is predominantly found in bone marrow. That was a single area where we had. But now we have discovered that hemoglobin bodies, which are called as or which are found in chondrocytes, are one of the important sources also. So which is recently discovered. Clear? Fine. So with this discovery, there is other researches which are moving on with respect to hemoglobin and functions of hemoglobin, how these chondrocytes impact the formation of cartilage. So if chondrocytes are important in formation of cartilage, then automatically hemoglobin is also important in formation of cartilages and so on. So consequential developments were there. Okay. So in this discovery, as a part of it, we had another important development what we had with respect to India. Definitely this time sickle cell disease is very important. So they can ask you because many developments are happening for past 1 or 2 years. Okay, we have special schemes addressing this particular disease. Apart from that, we also have discovered many therapies. That is what we call it as a Casgevy therapy, where we try to use gene editing technology to deal with this particular thing. Okay, for that only even Nobel Prize was there in 2020. So this particular sickle cell disease, which is one of the inherited disease, it is a genetic disorder where the RBC in the hemoglobin, which carries the oxygen, have some deformities. So that is, it will take a C-shaped form. So in which what happens, that is sickle shape. Okay. So the bonding with oxygen is inhibited in this particular ways, thereby it enters into various problems of anemia. Okay, chronic anemia will be there. So sickle cell crisis will be there, bones and chest. So arms, legs, in various area we have intense pain. So delayed hormonal growth, everything is problematic in this particular aspect. Okay. So what they do, they usually do blood transfusion so that the anemic condition will not be happening, or temporarily it is done away with. So in this particular case, what happened to treat sickle cell disease there is a therapy which is a gene editing therapy which was introduced. Okay. So in this particular case, what happens? They try to have it as a one-time treatment. What is one-time treatment? So once for all, it will be cured in case if you get that particular therapy. So how it is done? Usually, there is a gene which is named as BCL11A, which is actually one of the most important gene where this particular therapy actually targets. Okay, because this particular gene is switching from the fetal to the adult hemoglobin, where this particular thing is targeted. So by targeting it, we are trying to do away with that particular disease, transmitting from one to the other. So in this particular case, what they do is they try to edit that particular gene and they try to modify it, and then they inject it in the same patient. That is why we try to get that particular same blood cell, that is own blood cell. We try to edit it and we try to supplement it and provide it. So for that, we try to have the CRISPR technology. This is a one which the molecular scissor, or we call it as gene editing, which we actually do. And you know how the gene is being edited. That is, the infected area you will be having, and that particular gene will be cut off, and the replacement gene will be there, and that particular replaced gene, the defective gene, will be cured, and the original gene will be placed. But the problem here is this technology, though it shows that it can be possible by editing the DNA and replacing the faulty hemoglobin gene with a healthy one, and you try to supplement it to the patient, and that particular disorder is done away with. But the problem here is it is highly costlier. So it may cost around $2 million per patient. Okay. But anyway, this is the first step what they try to have. So a genetic disorder treated at the genetic level itself, we try to have that particular disorder done away with in this particular case with gene editing technology. Okay. So all three have relative aspect, but three are the different discoveries what we have made in different time period, but all three are related to each other. The news, I am talking about the news which are related to each other. Clear? Okay. So same way, we have another technology which is now the Indian authority, like the Central Drug Standards Control Organization, they tried to approve for its trials in India. This we call it as the CAR-T cell technology, or chimeric antigen receptor T cell technology, where this T cell is targeted. What are T cells? Popularly T cells are related to immunity. Okay, immunity cells. So many diseases they target T cells, especially HIV-like syndromes. So diseases they try to attack these T cells. So here also what they try to do, they target these T cells, especially for diseases like leukemia. Okay, so for these cancerous diseases, what they try to do, they try to get the patient treated for cancer using their own cells. Okay, so using their own cells. So how they do, they try to find out a gene. Say what cancer does is they try to inhibit this particular T cell so that it won't react against this particular cancerous growth. So we try to target that particular gene using our own T cell gene, and we try to have that particular modified cells, which is called as a living drug, because you have altered that particular cell, that is of the patient. You try to re-inject it, and we try to use them as a new T cell which is going to attack these cancer cells. So in this particular method, what they try to do is that the protein, okay, the protein which is actually there on the cancer, in this particular gene, so that binds to the patient's cancer cells, and this particular modified cell tries to kill this particular cancer cell, and that turns to be a memory, right? So we know that T cells have memory cells. So that will turn into a memory whenever the continuous cancer cells generate. These living drugs will try to put off this particular generation of cancer cells, and this is how leukemia, which is a popular cancer emanating from white blood cells. So this can be cured, okay, which inhibits that particular immunity. So this is one cell therapy what we have. Another similar method what they had is a viral treatment. So they identified viruses which can target these cancer cells. So injecting these virus into the cancer, patient, they try to target that particular cancer cells and it can diffuse that particular cell. So those type of virus, from that virus protein, it is being taken and it is like vaccine. So you try to fix that particular virus, a part of virus in that particular blood cells and you inject it. That is another separate method, but is also in current affairs. They have tested against cancer. So using viral protein, so against cancer. So we try to use our own T cells, so regenerate the T cells. So that can also retard that particular cancer. That is CAR T cell therapy. Whereas viral protein can also be used. That is another therapy which has been adopted. Here, why it is important? Because IIT Bombay, so that incubated a startup and that particular startup has come up with this particular treatment. So it is important, right? So anyway, but this CAR T cell therapy, it's not that complete, free from issues, but naturally, since we are modifying certain things, there are issues what we have in this regard. Especially we have neurological symptoms. There may be potential side effects, which they say because of the changes, what we do it in the protein and so on. But anyway, so they find it as one of the important area where we could easily do this particular what do you call the treatment, okay? So which will be very helpful for many cancer patients who are there in India. Fine. So this is one. So in this, the most importantly, either they can ask you that particular T cells, or this is the area where you have the questions. Okay. So what are the blood cells we have? So we have three types of blood cells. We have platelets, RBCs, white blood cells. So apart from that, you have the serum where you have the water body, what we have. So in this platelets, you have thrombocytes which help in blood clotting. Okay, you know that. So in case of any thrombosis or something, so blood clotting may be inhibited. So you know fibrinogen. So those aspects. So you know thrombocytes or thrombocrisis and so on. Thrombosis kind of ailment. So those issue you would have known. And similarly, this particular vitamin K, which is helpful in this. So that also you know that. And apart from that, try to also have, say, some disorders, some diseases, which brings down the platelet count. So naturally that results in fall of blood clotting or blood thrombocytes count will come down. So naturally that will create a major problem in human body. So these are some aspects what we have in this regard. Apart from that, we have red blood cells. Okay. So red blood cells, erythrocytes, which carry oxygen. Okay. Erythrocyte is the one which is linked to what you call the iron. Okay. Ferric. And apart from that, we have white cells. In white cells, there are different types of cells. So this lymphocytes is the one where we are talking about in this particular CAR T cells. So which carries T cells. Okay. So this T cells triggers immune system, and neutrophil is the first line of defense. After that, you have lymphocytes. Eosinophil, that is eosinophil, is nothing but it is a thing which is allergic response. Basophil as well as eosinophil. And monocyte is the one which breaks down bacteria and cleans up damaged cells. So all these functions are white blood cells function. So response to allergy, line of defense, triggering immune system, breaking down of bacteria, all these things are done by this particular aspect. Clear? So this is more important where your question lies here. So these fundamentals have to be understood. Clear? So which are they can even match what type of diseases affects what type of issues. I mean cells. So they can ask you in that particular mode also. Clear? Fine. So now we move on to the funga. So what is funga? Why it is in news?
So I'm not sure which body or which institution came up with it. I think it is IUCN, but like we have flora and fauna. So we also should have a repository and information about the funga. That is the concept, I think. Biodiversity.
Okay.
Yes, sir.
So UN biodiversity, and as you mentioned, IUCN also had an organization. We have species
Special
survival commission.
Survival Commission, right? Yes, sir.
So both have, I mean, this organization has first come out with the funga, importance of funga. So that is mycological inclusion, they say. So we see only flora and fauna. So flora is plant and fauna is animal. So including we need to have funga. So that's how they come up with. So first it was mooted by IUCN and later UN biodiversity also recognized it, and they said along with this will be there. So okay, so in funga, what are all the categories we have, or what type we have? What are all the classifications, or what organisms will be included here? Types. Say for example, mushrooms. Then
So yeast.
yeast, molds, right? Yeast, molds, all these come under this particular category. Okay, fine. So whether they are eukaryotic or prokaryotic, any idea?
So eukaryotic.
Eukaryotic, that means they are eukaryotic. That means what?
The
Distinct
nuclear
nucleus.
membranes and
Ah,
also are
so they
not
have
covered.
Nucleus is
nucleus.
covered with nuclear membrane.
So they have nucleus, cell organelles, everything. Okay. Fine. Prokaryotic means pro is something prototype kind of. So it may not have. Whereas here eukaryotic. So it is having. Okay. So whether how to put it? Whether these fungi play a role in carbon sequestration. Carbon storage, carbon sequestration. Say it will be. Why? Because fungi plays a major
They
role
have-
in nutrient cycle as well as carbon cycle, right? So in both the cycles, they play a major role because they are decomposers, right? They are decomposers. So naturally, they will have a role to play in carbon storage as well as carbon crisis. I mean, sequestration. Okay, fine. So apart from that, there are fungi or funga which can be used as a bio controllers. Same way, there are funga which acts as diseases also, diseases causing agents. So both the good part as well as the bad part are there with these guys. So it can be a toxin, It can be a disease or pathogen. It can be affecting crop, plant, animals, everything. So in one set. The other set, it can be also a biocontroller. It can be a biofertilizer. It can be used as biofertilizers. Can be. Okay, fine. So these are some aspects which you need to remember. And apart from that, fungi, they release mycotoxin that you can remember. So mycotoxin area. And whether fungi degrades pollutants, whether it has the ability to degrade pollutants like plastics, petroproducts, pharmaceutical products, oil.
So as they can act as a decomposer, so they can.
So it is possible, right? Okay. So it is possible. Fine. So these are some of the important aspects which you need to remember with fungi. So otherwise, they'll be going with the basic aspect what they are. And sometimes they say, remember, fungi can act as parasitic as well as symbiotic. So symbiotic relation can also be there because this is what it is. They have both positive side as well as negative side, right? So they can act as symbiotic relation. They can be parasitic relation. They have biocontroller relation. They also have the harmful effects, which can be there for human, animal, any plants or trees. Anything can be there. Clear. So it can be always in all aspects they are possible. Okay. So that's the reason why they try to put them under what do you call a separate thing called flora and fauna. Likewise, they try to have one thing. So what are saprophytes? What are saprophytes?
Decomposers are generally known as saprophytes.
So decomposers are called as saprophytes. Okay. So fine. Okay. So what is heterotroph? Heterotroph.
So they're organisms.
Organism that eats another plant and animal.
Okay. So heterotroph means fungi is an heterotroph because they try to consume the other organism for nutrients. Okay. Autotroph means plants. So they try to generate their own energy. But here for energy, they need to depend upon other organism. That is the reason why they are saprophytes also. Clear? Okay. Fine. So these are some of the areas where you need to have. So whether fungi will be having the potentiality of photosynthesis?
No.
No, because they are
No, sir.
heterotrophs. Okay, because they
Heterotroph.
are heterotrophs. Okay, fine. Whether fungi has ability to ferment?
Yes, sir.
Yes,
Yes.
because they consist of yeast and molds, right? So definitely they can involve in fermentation. Okay, fermentation process, it is easy. Okay. Whether they can be used in medication?
Yes.
It is possible. Antibiotic
Yes,
medication,
sir.
we can have these guys. Okay, so these are some of the areas where you need to know about fungi, right? So apart from that, there are some areas, minor areas, where you can see what do you call the xenobots, okay, quantum engine. Okay, so likewise, there are some other topics also related to genetic engineering. Okay, Y chromosomes. So these are basics which you can have the definitions alone. Really, you have some basics about Y chromosomes, right? So what is the unique characteristics of Y chromosome? Y chromosome deficient, Y chromosome linked disorders. So Y chromosome linked disorders are something fatal. Fatal in the sense it's very difficult to be rectified kind of. Okay, so those type of questions are minor issues. So you can just have a look upon. Clear? So these areas. Apart from that, not much are there. See, these are certain areas which are very nuanced, which we have discussed, which are favorite to UPSC also. Okay, so these area you focus on. Fine. Okay. So
Yeah.
apart from that, the second set of science and technology when it comes with 2024, we will discuss it again with relation to that. Okay. So these are some of the important areas which were there in 2023. So in 2024, we will discuss the Jan to April. At that time, we will see some more science and technology questions. Okay, fine.